Okay, so this is part 2 of the chapter 21 lecture notes. Let's see, let me put it back to the slideshow presentation. Okay, and sorry. There we go. Okay, so the last slide that we were looking at was this one here, that the one on the HIV retrovirus. Um, a retrovirus has an RNA nucleic acid. So the nucleic acid core is RNA instead of DNA. And so what happens is reverse transcriptase is also contained within the core of the virus, and it can take RNA and transcribe it into DNA. And that's why it's called reverse transcriptase, because transcription is normally the other way around. It's normally DNA is transcribed into messenger RNA. So this virus causes AIDS, acquired immunodeficiency syndrome, and it is an enveloped virus. So two things that kind of make it difficult to make a vaccine for and to treat is because of the fact that it's a retrovirus and also that it's enveloped. So viruses have a genetic component. All viruses have a core of a nucleic acid, and it can be DNA or RNA, but not both. And the DNA, and this is what is different from Bio 111, we learned that DNA is always double-stranded and RNA is always single-stranded, but in viruses, DNA can be single-stranded and RNA can be double-stranded. So that's one of the ways that we classify viruses, is by what type of nucleic acid is in the core, either single or double-stranded DNA or single or double-stranded RNA. The nucleic acid may be circular or it may be linear, and it may be in one piece or it may be in multiple segments. The influenza virus, for example, is in multiple segments, which makes it hard to develop a vaccine that's effective for the influenza virus more than just for one season of the flu. The DNA in viruses that have a DNA core, the DNA directs the host cell to make new virus copies. And then if the core is RNA, sometimes that RNA can be directly transcribed into messenger RNA, and sometimes transcription is not even necessary because the RNA already functions as messenger RNA. But RNA viruses are different from DNA viruses a little bit, because RNA viruses use enzymes that make more errors and they mutate more frequently, which makes them more difficult to treat. There are several ways to classify viruses. One way is by nucleic acid type, either single or double-stranded DNA or RNA. Another way is by the structure of the capsid. We talked about helical, icosahedral, and complex. And then whether the virus is enveloped or non-enveloped, and the structure of the genome, whether it's linear or circular. And these are some different classifications of viruses. Some DNA viruses are rabies, and some retroviruses are DNA viruses. Not all of them, though. A herpes virus that causes herpes and also cold sores is an RNA virus. Smallpox is an RNA virus. You can also look at single-stranded and double-stranded viruses, and linear versus circular, and you can look at non-segmented versus segmented. This is a way of classifying viruses by the structure of the capsid. You can have, for example, if it's called naked, there's no envelope. And if it's called enveloped, then it has an envelope outside of the capsid. So an example of a naked helical virus is the first virus ever discovered, which is the TMV, or tobacco mosaic virus. And enveloped helical would be just like that, except it has an envelope outside of the capsid. And the influenza viruses, mumps, measles, and rabies, these are enveloped helical viruses. So these are just different classifications based on the structure of the capsid. Now, the Baltimore classification is more specific about the nucleic acid core. And using the Baltimore classification, there are 7 groups of viruses. The first group are double-stranded DNA viruses. And if a virus has double-stranded DNA, then transcription and translation happen exactly as they happened the way that we learned in human cells, for example. DNA is transcribed into RNA, into messenger RNA, that's transcription. And then messenger RNA codes for the amino acids in the protein. So that's translation. So a double-stranded DNA virus carries out transcription and translation just like we've learned. And another thing a virus has to do is replicate its nucleic acid core. So a virus is going to have 2 jobs. One is to make the protein through transcription and translation, and the other is to replicate whatever its nucleic acid core is, because a new virus is a combination of the proteins that form the capsid and the nucleic acid core. So an example of a double-stranded DNA virus is herpes. An example of a single-stranded DNA virus, which is class 2, is parvovirus, which causes parvo in most of the time that happens in puppies, not older dogs. But the only difference is that the single-stranded DNA has to be converted. It has to replicate and form double-stranded DNA before transcription and translation can occur. And then that single-stranded DNA is replicated. Class 3 is double-stranded RNA, and in that case, transcription occurs by using the double-stranded RNA genome to transcribe messenger RNA, and then translation happens. So, and then the double-stranded RNA molecule has to replicate, of course. So childhood gastroenteritis is an example of that, and that's a stomach and intestinal type problems, also called rotavirus. Class 4 and 5 are single-stranded RNA viruses, but class 4 is single-stranded RNA positive virus. And in that case, the single-stranded RNA is the messenger RNA. So there is no transcription in class 4 viruses. The common cold is a class 4 virus. It's called Hercornavirus. And in the single-stranded RNA negative viruses, messenger RNA is transcribed from the RNA genome, and then translation occurs. So there's one extra step in the class 5 viruses than there is in the class 4 viruses. Rabies is a class 5 virus. Single-stranded RNA viruses with reverse transcriptase, one of those is HIV, human immunodeficiency virus. And then class 7 is where the genome is double-stranded DNA, but the replication of the viral DNA is more complex than in class 1. So hepatitis B is an example of a double-stranded DNA virus that also contains reverse transcriptase. And you can watch this video link to show how the flu virus invades your body. I'm not going to play the whole video, but I do want to show you when you go to it, you kind of have to scroll down. I couldn't find it at first, but you read a little bit about the flu virus first, and then you keep scrolling down, and it'll say web resources. Sorry, I went too far. watch the original animation by David Belinsky and his team, and that is the video that it's talking about. Okay. Oh. Okay. Um, so the effects of viruses on cells are called cytopathic effects. This is when cells are damaged because of the effect that the virus has on the cell. Sometimes cells are damaged to the point that they're destroyed, and it causes, sometimes when the virus replicates inside cells, when there are so many viruses that replicate, when they burst through the cell, the plasma membrane of the cell, it causes the cell to rupture or lyse. And sometimes viruses have an effect on a cell in which they cause apoptosis or program self-destruction of the cell. And there are other symptoms that are caused by cell damage and also by the way the body's immune system responds to the virus. We have symptoms of illness that's just simply the inflammatory response, such as fever, um, you know, redness and swelling and things like that. In some cases, replicated viruses can leave the cell by budding. They can kind of form a little bud off the plasma membrane. And I was going to see, there's a picture somewhere, but if this is the host cell, for example, a little piece of the host cell can kind of bud, and let's say the virus is this little dot. So that little piece can actually eventually break off. In fact, when that happens, the piece of the plasma membrane that broke off of the cell is the envelope. It forms the envelope for the virus. Steps of virus infections are attachment, entry, replication, and assembly, and then a step called egress or release. In the attachment step, the virus has some type of surface proteins, sometimes glycoproteins or the capsid proteins, but the virus has these particular proteins on its surface that must bind to receptors on the host cell. So a virus can only infect a host cell if it has surface proteins that can bind to receptors on the host cell. And that makes viruses very specific about which cell they infect. Then there's entry. And viruses can either enter just by injecting their nucleic acid into the host cell, which is how bacteriophages work. The bacteriophage DNA just enters naked. In other words, the rest of the bacteriophage, the capsid that's shaped kind of like a spaceship, remains outside of the bacterial cell that it's infecting. And sometimes the entire virus can enter the cell. It can enter by endocytosis, or, so here is endocytosis, where the cell is just engulfing the virus. But an enveloped virus can actually fuse with the plasma membrane, and it's still like the same thing, but it's just that the envelope fuses with the plasma membrane because the envelope and the plasma membrane are both composed of a phospholipid bilayer. So sometimes the entire virus enters the cell, and sometimes it's just the genome. Okay, so there's attachment, there's entry, and then there's replication and assembly. The viral genome has to do two things. It has to replicate so that a new virus can have the genetic material as well. And then it also has to carry out transcription and translation to make the proteins that form the capsid. So if the genome is DNA, then it will transcribe messenger RNA and make proteins through translation, and then it will replicate or duplicate the DNA. So that's the components of the virus is DNA and the protein capsid. And other viruses will replicate and assemble in different ways depending on what the viral genome is, whether it's DNA or RNA, or whether it's a retrovirus or not. But all viruses have to replicate